I hope you enjoy this at Yaaka, and I hope you find us worthy of you subscribing to that be happening regularly. Mute your mic and tap on it to mute. And tap on it. So
Okay. Uncle Ombuta Yombe Soduga.
all right. So today we shall look at cell biology. Is one of the first topics that you always look at in S5. I felt like it was very, very important for us to go over it. Please, if my internet breaks or if you can't hear me well, you can type in the chat box or you let me know. And I see what to do. Okay.
Like want to talk to you. Yes.
Faith, your mic is on.
Okay, actually I'm in class. Let me get done, then I get to you.
I've muted her. You can start.
Yes, teacher, I'm sorry.
Okay, it is fine. It's very okay. Please, if my internet is bad, you type in the chat box. I'll see what to do.
Okay.
All right, so today we'll be concentrating on cell biology, and I'm glad to see all of you here. I hope you enjoyed this lesson. I hope you enjoyed the entire experience on Yaaka as you're here. Okay, so in cell biology, we'll just be looking at ideas about the cell structure and how they have changed considerably over years. But we see that early biologists saw cells simple membranous sacs containing fluid and a few floating particles. Today's biologists know that cells are definitely more complex than this. So we've seen an evolution of how cells were regarded from the early ages to what they are right now. Right now, we see that they're so complex, more than the way they were seen a few ages ago. And that evolution has happened because of science, because of research, and so many other aspects. Also, this is an example of the variety and diversity of cells. And I would like someone to tell me which type of cells are shown in this picture. Can someone tell me? You can just unmute your audio and say it. What type of cells?
Erythrocytes.
Okay, that is correct. These are red blood cells or erythrocytes. Well done, Faith. So we shall look at a variety of them, but first of all, we'll first concentrate on the prokaryotic cells. Prokaryotic cells. Here is an ultrastructure of a prokaryotic cell. This is a rod-shaped bacteria. There's so many species of rod-shaped bacteria, but these are the common features that we have in them. Okay. When you look at a rod-shaped bacteria, one of the distinctive feature is the rod shape. This is a rod shape here. Okay, the way it looks like, it's like a rod. This is why we call it a rod-shaped bacteria. The other distinctive feature of most bacteria is the circular DNA that is wound in the cell, which is here. We call it the nucleotide. It's their DNA. It is not found in an enclosure. So therefore, prokaryotic cells do not have a nucleus. As you can see in the word prokaryotic, there is a word pro, which means before, and karyon, that means the nucleus. So these are called before the nucleus, meaning they do not have a nucleus. Okay. Their DNA is not covered in a double membrane or an enclosure. That is a distinctive feature. We see a flagella or a flagellum. It's another distinctive feature which they use for what? Who can tell me what the flagella is used for in the prokaryotic cells? Unmute your audio and say it. Who knows? Hamza, do you know? Yeah? Okay. We shall look at it, but mainly it is movement. Okay, it's movement. Another distinctive feature that we have, is the circular, the plasmid. These plasmids are just small DNAs that are found in the bacteria. Okay. Small DNAs that are found in the bacteria. Their main job is seen mostly in their division, their cell division. We have a cell wall. It's another distinctive feature here. Cell wall. We have a capsule. We have pili, ribosomes, very good for protein synthesis, cytoplasm, of course, and then a plasma membrane. Those are the key features of a prokaryotic cell. Please, if you have questions, you ask. Just unmute your audio and ask. Or you can raise your hand and ask. Okay. So that is the structure of a prokaryotic cell. We also see this guy here. Biologists, instead of taking selfies, they take selfies. Okay, especially like selfies. So I hope you remember that. My internet is not good. I hope it's better now. Is it better?
Yes, teacher.
Okay, good. So we're going to look at the parts present in the parts that are present in prokaryotic cells. Bonita, your audio is very loud. Bonita, your audio.
I'm on.
Okay, yes, you are on. We can hear you loud and clear, but unmute your audio, please.
I'm on, but I'm not getting you.
You're getting what? You said you're getting what? Okay, I guess the audio has cleared a little bit. Can you guys still hear me? Can we
Yes,
continue?
you are loud and clear. Continue.
Okay, good. So we're looking at the parts that are present in the prokaryotic cell and some of their importances. We have a 70S ribosome. The 70 represents the type of structure it has. So the 70S ribosome is a site for protein synthesis. We have the cell wall. The cell wall contains a peptidoglycan layer that protects and maintains cell shape. This peptidoglycan layer is also used to distinguish between different types of bacteria, as we will see. We have the cell membrane, which is a phospholipid layer which contains which controls entry and exit of substances. We have a nucleoid, which is a region of one free strand of DNA. We have food granules, which are glycogen and lipids for food storage. And then we have the cytoplasm, which is a center for biochemical reactions. These are the parts of a prokaryotic cell that are always present in any prokaryotic cells. So these ones, you always find them. The other parts that are not always common in every prokaryotic cell, and we shall look at them also. Here is another 3D structure of a prokaryotic cell that shows all the components of this cell. Here you can see them clearly, the single-stranded DNA, you can see the circular DNAs, you can see the different organelles, the ribosomes, the food granules everywhere. So it's a very good structure that shows most of these. If you have joined, please mute your audio. You just joined, mute your audio. Okay. Now let's look at the parts that are present sometimes. Okay, these are sometimes present. They're not always present, but only sometimes. These are, the first one, we have the mesosome. The mesosome is a site of respiration, but also it has a very important job of cell wall synthesis. Okay, we have the flagellum. It's elongated, relatively flexible, corkscrew-shaped structure that moves the cell. So its main function is movement. This is also sometimes present. We have the capsule, or what we call the slime layer. It's always for protection. We have the pili or fibri. These are protein filaments that facilitate cell adhesion and conjugation. Okay. These facilitate in the cell adhesion, attaching to various substances, but also they work in conjugation, where reproduction is involved. We have the plasmid, which is independent small cycle of DNA, which offers resistance to drugs. This one is very, very key in offering resistance to drugs to these bacteria. Photosynthetic membranes, where photosynthesis occurs. These are also sometimes present. They're not always present, but sometimes present in some prokaryotic cells. Okay. So those are the parts that are sometimes present in a prokaryotic cell. Please feel free to ask questions if you have any. If you have something you have not understood or you'd love to be explained more, please ask. Okay. This is another structure that shows a very, very good representation of a prokaryotic cell. You can see all the organelles well represented, and it's very, very good to look at. On our websites, we have videos and very nice interactive activities about these prokaryotic cells. If you sign up for Yaaka, you'll be able to access all those. We have some nice 3D videos also. So this is a very good structure also for a prokaryotic cell. Differences in cell wall structure. Now, this is a difference that we use to distinguish different forms of bacteria or prokaryotic cells. We have the gram-positive cells. These ones have a thick peptoglycan layer. Okay, this term here is very important to remember about gram-positive bacteria. They have a thick peptoglycan layer that reacts with the gram stain to form a violet complex. Okay, so when you stain bacteria, this is actually what picks up the color of that stain. For example, if you're using methylene blue, you'll see that violet complex being formed if the bacteria is gram-positive. If it is gram-negative, you won't see that because the gram-negative bacteria contain a thin peptoglycan layer. Okay, so therefore this cannot be stained by gram. Gram stain cannot work here in the gram-negative cells. Let us see what happens when you stain these, the different forms of bacteria. Okay, so here is a very nice structure that shows the difference in the thickness of the peptoglycan layer. Here is the difference. As you can see here, when you look at the peptoglycan layer, so right here, it is very, very, very thin. It is thin compared to this gram-positive one, which is very, very thick. Okay, as you can see, this is thick compared to the other one. Right? This is the peptoglycan layer in gram-positive. This is the peptoglycan layer in gram-negative. Here thin, here thick. That is the difference. So gram-positive can be stained. These ones cannot be stained by gram. Okay, by the gram stain. That's the difference. Okay. Let us see another example in staining these different cells. We can see here that we have the gram-positive. As you can see, the peptoglycan layer here, very, very, very thick. Okay, from here up to here. So therefore, when you use gram stain to stain a couple of the different bacteria, you see the gram-positive ones, they'll have this color, the violet color. Okay, so this will show that these are the gram-positive bacteria. The ones that are not stained, that do not give off the bright violet color, these, okay, like these, like these, those ones are gram-negative, gram-negative bacteria. Okay, so that's the difference. As you can see here in the gram-negative, the thickness is very small. So this is a thin peptoglycan layer, very thin compared to this huge one. Okay, and that's why the gram stain can stain gram positive ones, yet cannot stain the gram negative. The difference is in the thickness of the peptoglycan layer. Please ask questions if you have not understood or you are confused somewhere. Please ask. So that is the staining. Okay, that's how it occurs. And that's how we differentiate between these two forms of bacteria, gram positive and gram negative. If you have any questions, please do ask. Okay. Let's look at the comparison of eukaryotic and prokaryotic cells. So we have a feature here, then we shall have a eukaryotic cell, what it has, and then a prokaryotic cell, what it has. For example, let's start with examples of each. Examples of eukaryotic cells, we have cells of plants, animals, fungi, and protists. Examples of prokaryotic cells, we have bacteria and cyanobacteria. Okay. Let's look at more differences. This is a table showing the differences between each. For example, we can start with this, the cell size. Let's start with the cell size. Prokaryotic cells are much larger than eukaryotic cells. We see that eukaryotic cells are larger than prokaryotic cells. We see that the prokaryotic cells are much smaller. You can see the values in micrometers there. Cellularity, let's look at another feature here, cellularity. Usually, eukaryotic cells are multicellular, mostly unicellular for prokaryotic cells, and mostly unicellular. Some cyanobacteria are multicellular. We have some that are multicellular, since some of them are a combination of many. Another feature we can look at is a nucleus. A nucleus. It is present with a clear envelope. Okay, so here we have a nuclear envelope. Here we don't have a nucleus at all. Okay. Like we saw in our diagrams, in the prokaryotic cells, it is just DNA. It's just DNA present in the cell, single-stranded DNA present in the cell without a nucleus. Another feature we can look at is the shape of the DNA. Shape of the DNA. The DNA here is linear. Here it is circular, meaning it has no ends. It is circular DNA. Then the DNA composition is another feature you can look at. In eukaryotic cells, DNA complex with proteins called histones. It's complex DNA with proteins called histones. Here, DNA is naked, without histones. Okay, without histones. When you research the structures of DNA, you see what we mean by the histones, those proteins that are found on DNA. Main organelles, they are present here. Here we don't have organelles, the double membrane organelles. Ribosomes, here we have a much larger 80S ribosome, and also we have the 70S ribosome, the cytoplasm in eukaryotic cells. But in prokaryotic cells, they are only smaller ribosomes, which is the 70S type. Okay. Remember, the 70S represents its structure, which is this word here, the safe bag. we can look at the flagella or the flagellum. If present, it is a 9+2 microtubule arrangement, 9 peripheral doublets surrounded by 2 central singlets. This is a structure, this is a protein structure of the microtubules found in the flagellum. Here, if present, it lacks the 9+2 microtubule arrangement. So the microtubules here in the flagella found in the prokaryotic cells, it doesn't have this 9+2 arrangement of 9 microtubules. Okay. Microtubules are like thread-like structures. So the arrangement here, we have the 9 that are peripheral doubles, and then 2 central singlets. We have the cell wall. We have the cell wall. The cell wall here in the eukaryotic cells is chemically simpler. In plants, it is made up of cellulose. In fungus, it's made up of chitin. Animals have no cell wall. For the prokaryotic cells, the cell wall is usually chemically complexed with the peptoglycan layer that we just looked at in the staining, in the chrome stain. We have the plasma membrane, is another one. The plasma membrane, it is sterols and carbohydrates present, the plasma membrane. There are no carbohydrates and it lacks the sterols. Okay. We have the glycocalyx, is present here, present in the eukaryotic cells, okay, in some of them, especially those that lack a cell wall. Here it is present as a capsule. Sometimes we call it the slime layer. And then cytoplasm, you may not see this well, but we're talking about the cytoskeleton. Cytoskeleton are just a few microtubules found in the cytoplasm. So in the cytoplasm of eukaryotic cells, there is cytoskeleton, while in the cytoplasm of prokaryotic cells, there is no cytoskeleton. Like I said, the cytoskeleton are just microfilaments and tubules found in the cytoplasm. So those are the differences between eukaryotic cells and prokaryotic cells. I'll send out these notes when the lesson is done so that you can still go through it and see. So yeah, if you have questions about that, please remember to ask. You can always post questions in the groups so that I can answer them. But those are the differences. Okay, a structural illustration of some of what we have looked at is right here. As you can see, circular DNA found in the cytoplasm, which we don't have here. Our DNA here.
Excuse me, sir.
Yes, please.
Someone is raising a hand.
Okay, Bonita, please ask. Bonita, please ask your question. Yes, I can't hear you. If it's possible, type it in the chat. I'll see it. You can type it in the chat. I'll see it there. Okay, as I was saying, we can see the structural differences using this diagram also. You can see the nucleus here, which we don't have this side. It's no nucleus. We can see double membrane organelles, main organelles like the endoplasmic reticulum, the chloroplast, and many others which we don't have this side. We can see a capsule here which we don't have this side. You can see a complex cell wall which we don't have this side since this is an animal cell. And even if it was present this side, it wouldn't be as complex. Okay. Let's see this question. It's not necessary for you to write the 70S and the 80S, but it is important for you to know those structures. This is a 70S, meaning it's smaller, and this is the 80S, meaning it's bigger. Steroids are like fats, types of molecules that are under fats. Okay. That's a good question, Bonit. All right. So these are the visible ones that you can see. Structures. Just a good representation. We have a story here. So we have here a bacteria that was just moving about, and then it met a friend called penicillin. Bacteria was like, I guess I was going to do some cell division anyway. Actually, it was the penicillin that said first, hey, bruh, mind if I hang out for a bit? Penicillin was asking bacteria. And then bacteria was like, yeah, I guess I was going to do some division anyway. If you know something about penicillin, you just know what is about to happen. So penicillin interferes with the formation of the new cells, the new cell's cell wall. And it is like, whoops, sorry about that, bacterium. Because penicillin, if you used to have wounds a long time ago, I know they gave you penicillin or something. And penicillin, what it does, it interferes with the formation of the cell wall of cells. So therefore, the bacteria bursts due to the osmotic pressure. And then the penicillin goes on laughing seriously. Any problem, bacterium? That's a very short story of how bacteria ended up being destroyed by penicillin. Okay, I have an activity for you that you're going to do and submit in your group. Here is the activity. Explain the cell theory. Explain the cell theory. So write down your answers on a piece of paper or in your book, then take pictures and submit in the WhatsApp group. Okay, I'm giving you these few remaining minutes, like 15 minutes before we go for chemistry. To do this activity, I want to see it in the group submitted. Okay, any questions about this? If you have any questions, please ask before we go for this activity.
There's no question, sir.
Okay, so go on and do the activity and submit it in the WhatsApp group before we go for chemistry. Use the remaining 15 minutes to do this. Okay. All right. Lastly, can't tell if biology is a foreign language or it's just biology. Yeah, sometimes I feel like this too. There's so many new words. Sometimes it feels like you're talking a foreign language. And then we have my friends here. Every time the teacher says the next chapter is human reproduction, this is the face they always give when the teacher is about to talk about reproduction. Thank you for attending. I hope you subscribe for Yaaka more lessons, and please go to our websites and go through the courses that we have so that you can benefit from them. Please go do my activity. That's it for today.